चल रहा है। बंद करने की बोल रहे फोन करें। हाँ अब चलाई बोला है सबको बंद करना है। नहीं नहीं उनको बोल रहे थे अपना ये वो मत मिस कर। बोल अंकित को बोल देते ना उनको 15 मिनट बोलना यहाँ अगर खाली तो ठीक है।
everyone. Welcome to the 16th Annual Research Seminar. I also extend a warm welcome to our newly extended family of SACON who have joined us online. It's really nice to see this all filled again after a gap of 3 years. So, welcome to you all. I would like to request the dignitaries to kindly take their place on the dais. Our seminar chairpersons, Professor B.C. Chaudhary and Professor B.K. Unya, our director, sir, Dean Dr. Satyakumar sir, and our research coordinator, Dr. Bhutan sir. Sir, please take your place.
जूम करवा दो, जूम आउट करवा
जी
दो।
Our seminar chairpersons for the internal annual research seminar are Professor B.C. Chaudhary and Professor B.K. Unya. B.C. Chaudhary sir is very well known to all of us. He's also a very well known conservationist and herpetologist and a veteran expert. He's one of the first generation wildlife biologists of the country. He worked for almost 30 years with WI as a faculty and retired as scientist dean in 2012. He currently advises a large range of wildlife NGOs, including the Wildlife Trust of India, and is also an advisor to the Government of India. I now request our director to kindly felicitate B.C. Chaudhary sir. Professor B.K. Unya, sir is a 1982 batch IFS officer. He's retired as PCC of Kerala. He's been a faculty at WI for a very long time and has expertise on wildlife management. And his management plan written for Paramariculam continues to be one of the best management plans ever written and is still followed very thoroughly. He carried out research on high altitude grasslands and is known for his excellent work on eco development initiative. He continues to provide the much needed insight of translating science into management and continues to provide inputs in WIS courses. I now request our director to kindly felicitate Unyal sir. Thank you, sir. I now request our research coordinator, Dr. Bhutan to kindly deliver her welcome.
Thank you, Vishnu. Good morning to all. I welcome you all to the 16th Internal Annual Research Seminar. It is really great to meet again after a gap of 3 years after the pandemic. The 15th Internal Annual Research Seminar was held in 2019. This year, the IARS has 60 presentations scheduled in 11 sessions of 2.5 days. As you are all aware, the Sanivani Center for Ethology SECON is now a part of WII. On day 2, there is an online session of 16 presentations from SECON. I extend a warm welcome to our friends and colleagues from SECON who are joining online. The idea of Internal Annual Research Seminar was generated from the fact that in early 20s, we were growing and so were the number of projects, and therefore, since all the projects could not be accommodated in the annual research seminar, it was essential to review them. In 2005, we began the internal research seminar. The projects with more than 6 months duration and up to 2 years were given a platform as IRS to present their work. This is also a critical review process that helps the researcher in refining methodologies, analyzing and interpreting data, and get a feel of scientific presentation and answering to questions. This rigorous exercise helps the presenter to be confident and improve upon. Constructive criticism and suggestion should always be welcome, and I'm sure our presenters are geared up for it. I wish all the presenters all the best. The 2.5 days will be educative and fun-filled. I am looking forward to it. It is also my pleasure to welcome our seminar chairs, Sri B.C. Chaudhary and Sri B.K. Punyal. Thank you, sirs, for agreeing to be the chair of the internal annual research seminar. Thank you.
Thank you, ma'am. I now request our Dean, Dr. S. Satykumar, to kindly deliver his remarks.
Thank you, Dr. Studia. Very good morning to all of you. Seminar chairpersons, Professor B.C. Chaudhary and Sri B.K. Punyal, Director Sri Virendra Tiwari, Dr. Tapisina, Research Coordinator, my colleagues, faculty colleagues, colleagues from SACON who have joined online, all the researchers, students, and ladies and gentlemen. It's a privilege to be here. As Dr. Mitapi just mentioned, it is an occasion that we all should be happy about, that we are here back doing the annual event, that is our ARS after the gap of 3 years. And I would like to tell you that this is an annual event that every researcher, student, and faculty looks forward to. And we have a very strong history to what we have been doing all these years, and why are we enjoying all these years, and why do we really want to do this every year? Way back in 1985-86, the annual research seminar started in WII in a one-room office of FRI with just a dozen research scholars then. Later on, as we grew, we had to go into an annual research seminar that was about 3 days. And then we knew that we are having much more projects and researchers, we had to break it to IARS and IARS. And some of you may also know that in the last few years, before the pandemic, we even had seminars only for Campa projects on one day, Himalaya seminar for a day, and so on and so forth. So the IARS does not include the Himalaya seminars and the Campa seminars. So the growth, leaps and bounds that we had gone through today is that we have over about 700 researchers and doing projects. And if each and everyone wants to make a presentation, you can imagine the time that we will need to do this. The genesis of these, the seminar itself is that earlier all our projects were funded by granting aid. So the Training Research Academic Committee, then also known as RAC, wanted to review the projects. So we had started this annual research seminar every year so that the RAC members could see how the projects are performing and make a review of what was going on. And today, as you see, that in the last few years there are no granting aid projects. All the projects that we do are funded by various resources, sources, funding agencies, including the Ministry of Environment, Forest and Climate Change. Having said that, I would like to say a few things. The science we do has always helped us to build our knowledge on wildlife sciences in general, and specifically to address issues of controlling illegal wildlife trade, trying to help in improving the wildlife management in the country, and most importantly, dealing with the emerging issues of diseases as well as the human wildlife conflicts. Our science should help the managers and this country to utilize that information for managing the problems, the day-to-day problems that we are facing to save wildlife, the habitats, and also reduce human wildlife conflicts. So the information that we generate should be for a purpose, even if it is totally an academic research. By gaining knowledge on a particular species, not today, maybe 10 years or 20 years later, that information will be of immense use to the country, to all of us, and the managers who are protecting these species and habitats. This particular forum gives an opportunity to the young men and women here to make a presentation about what they have done. Doing good work is one thing, but not able to tell this to a forum is not good. So we in WI believe that our researchers should be given training in making presentations, and what we are going to do this week and next week is an example, and what all we have done for all these years is an example of how people should learn how to do good real good field work, collect proper data, analyze them very well, and make presentations. Presentation skills are extremely important for all of us. Present the work that you have done to any target group. Today it's an informed audience, but tomorrow you may have to speak to people who don't know about wildlife. In that case, your presentation will change, but you will communicate the findings in a simpler manner to reach the target audience. So, I will request all the presenters to make the best use of this opportunity to make your presentations. All of us are here for one reason, that is to conserve wildlife in this country and elsewhere in the world. And we are all connected by one thing, that is passion for wildlife. And that passion should be revealed to the audience when somebody makes a presentation. So this would help everyone to understand what one person has done and how this work is presented and how this work is defended is also extremely important. So I request all the presenters to make their presentations in full passion, and I'll expect all the listeners to be here in full attendance and do not walk away when somebody is making a presentation. And make interventions. Lively interactions will enable us to grow together. Like Dr. Bitapi mentions, critical or constructive criticism is more important. And there are some questions that you can ask here. Some you can always ask when you're having a cup of tea to the presenter who made a presentation. So I look forward to the next 2.5 days as the time period that when we all can have healthy interactions and learn more about from each other. And so wish you all a very happy learning exercise and looking forward to all the presenters, presentations and all the audience making interventions wherever necessary. Happy learning. Thank you very much.
Thank you, sir. I now request our director, Shri Virendra Tiwari, to kindly deliver his inaugural address.
Good morning to all. Chair for today's IERS, Professor V.C. Chaudhary sir, Shri V.K. Uniyal sir, Research Coordinator Dr. Bitapi Sinha, Registrar Dr. Satya Kumar sir, my faculty colleagues, all my researchers, and my faculty colleague and researchers from SECON who have joined online. So welcome to all of you. So we are meeting after a gap of 3 years for this ARS. I am given to understand that ARS started in 1986 and IERS in 2005. And due to COVID, we could not hold this research seminar in last 3 years. And as already told, we have about 60 presentations, and I'm around 20 plus poster presentation will be there. And it's a really privilege to be here addressing you all in presence of all our esteemed chairpersons and all learned faculty colleagues. So as already Dr. Satyakumar said, it's an opportunity to showcase your work, to internally here in IRS, and also when we have our ARS on 21st, 22nd, to other stakeholders, particularly forest departments. Now we had been doing research in various fields. Already Dr. Satyakumar mentioned that it should be relevant. There has to be a long-term research, and also we should have a medium term and a short term which solves the immediate problem which the nation is facing, particularly related to human-wildlife interface. So WI is, you all know, is a premium research institute. So far, we have about 190 plus PhDs awarded from this institute, and at present, about 158 PhDs are registered, and it's ongoing. We had been publishing in journals with good impact factor, and some of our researchers made presentation in international forum in various workshops and symposiums. So we are looking forward to more and more of you creating an impact internationally and of course in our country as well. We here in WI is trying to facilitate your work, provide better facilities to you, be it a hostel facility, be it a reading room facility. We are trying to improve. Soon you will find some visible improvement. We have also proposed a new hostel with a capacity of about 80 rooms. We have given consultancy to CPWD. They are appointing a consultant to design the hostel. And also one more thing, I just want to share. You'll be happy to know that we are preparing to launch a journal of wildlife science very soon. And maybe within a month, we are trying to launch it in first week of October at the hand of Honorable Minister during Wildlife Week. So preparations are in full swing, and maybe something which I was told we had been waiting for long to launch a wildlife journal in our country. So that I think it will be a very proud moment for WI. We'll be launching it, and maybe we plan to have the first edition in the month of March or April 24th. That will be the first. So I once again looking forward to. I intend to sit through and maybe learn some research from you what's going on. So I'm the only odd man out here with all researchers and scientists. So maybe I'll try to take this opportunity and learn about some of the work, interesting work which we are doing. And thank you so much. Thank you so much again. First of all, to Dr. Pidapi, who is our research coordinator, in organizing this IRS. I never knew it takes so much of work. So many of our faculty members are involved in the form of various committees, and preparing for the ARS. Some inconvenience may be tolerated. Last moment, the AC wasn't working. We are trying to sort it out. Maybe partly it is sorted out by evening. I think everything should be okay. Thank you so much. Looking forward to interaction with you. Thank you.
Thank you, sir, for setting the stage for the internal annual research seminar. I now request you to kindly take your place off the dais. We will start with our technical sessions. Just a housekeeping announcement for those of you who are presenting. Kindly identify your session facilitator and hand over your presentations and introductions if you have not already done it in the morning. And for those of you who are going to have presentations tomorrow, kindly ensure that you are here by 8:30 in the morning and you upload your presentations. We have a session facilitator this time for the internal annual research seminar, who's responsible for taking over the questions. There are question sheets printed, and you can write your questions on that, and that will be moderated and read out at the end of each session. So please, at the end of each of the tables, there are printed slips, and you can use that to write down your questions. If you don't have one, please raise your hand or identify one of the technical staff. They will hand over a question sheet. We will start our first technical session. It is on carnivore ecology. The session will be chaired by Professor Kamal Qureshi and co-chaired by Dr. Salvador Lingo, and the session facilitator will be Dr. Shikarish. I hand it over to the session.
Thank you, Dr. Vishnu. I like to start the session now. I thank WI for providing the opportunity of being the session chairman for today's proceedings, and it is my privilege to welcome you all to this session on carnivore ecology. This topic holds profound significance in our changing world as it delves into the engineering dimension of humans and implementing the environment in the environment. And it's through the study of carnivore ecology is the testament to our collective responsibility as a steward of the national world. It falls upon us to be both observer and custodian to understand the intricate balance that sustain ecosystem and the appreciate vital role that carnivore play within this system. I invite now Rivan Singh. He's going to talk about connecting the clouds, the analysis of cloud network, connectivity across distribution range in Northeast India. Over to you.
Tribhuvan Singh has done his master's from in environmental science from June University and joined the institute in 2021. He's currently working in Pan India Assessment of Cloud Leopard project.
Thank you, Dr. Brish. Good morning, everyone. I'll be presenting findings from connectivity analysis of covered leopards in its distribution range in Northeast India. Covered leopard is a medium-sized pellet which has a known distribution in Southeast Asia across 11 nations. And in India, it is found in 8 northeastern states as well as in the northern part of West Bengal. This is our study area, and these are the reference points we gathered from the literature community. Owing to the habitat loss and degradation and poaching, clouded leopard is categorized as vulnerable in IUCN Red List and listed as Schedule 1 species in Wildlife Protection Act and Appendix 1 of CITES. According to a recent assessment by IUCN, it was observed that the population of covered leopard has witnessed a decline of around 32% from 1999 to 2019. The objectives of this study are to assess the potential suitable habitats for the clouded leopards and to analyze the habitat connectivity among the core habitats of clouded leopard in India. But what is habitat suitability? Habitat suitability is a degree to which a specific area provides necessary environmental conditions and resources which are suitable for the survival and proliferation of any particular species. So we use spatial distribution modeling using Maxent, which expresses probability distribution of predator suitability in each of the grid cells in the landscape using occurrence points and environmental variables. And what is connectivity? Connectivity is the degree to which different populations and habitats are connected to each other. And it is important because connectivity ensures the gene flow and dispersal and movement of the animals from one population to another population. And if the connectivity is lost, then this isolated population becomes more vulnerable to local extinction due to inbreeding depression and diseases and various other factors. We use linkage mapper to map the connectivity among the core habitats. It works on least cost path analysis. Least cost path calculates the most cost efficient route for the movement of animal from one core to another core. It further integrates circuitscape to analyze the connectivity. Circuitscape is based on circuit theory, which considers a landscape as a electric circuit, and the current is represented by the movement of animal, nodes as habitat cores, and just as an electrical circuit has some components that resist the flow of current, the landscape also has some features that resist the movement of animals, such as physical barriers, rivers, mountains, and roads, and etc. So one of the most important steps in the connectivity analysis is to construct a resistance surface. So resistance surface is simply the spatial representation of movement cost across the landscape. It assigns the resistance value to each of the grid cell, which represents how difficult it is for an animal to move across that area. We use three of the most important factors which affect the habitat connectivity and movement of animals. Those are land use land cover, human population density, and transportation network. This is the final resistance surface we constructed. As it is evident that Brahmaputra River is acting as a natural barrier for the movement of clouded leopards, and these are the resistance scores we assign to the data layers and each classes of those layers. This is an overview of our methodology. First, we selected 57 environmental variables and 150 occurrence points. Then we removed highly correlated variables and verified the presence location to avoid clustering. And we ran the habitat suitability model using Maxent. And from the output results, we extracted core habitats of clouded leopards from the high suitability area with an area of 100 square kilometers. In the second step, we constructed a resistance layer using Nagli toolbox in ArcGIS, and the final resistance surface and core habitat layer were used as input for the connectivity analysis, where we use least cost pathways, centrality, and pinch points. According to our habitat suitability results, around 7% of the total landscape was under high suitability for clouded leopards, of which Arunachal Pradesh had the largest area under high suitability class. We also identified 30 core habitats which had an area of more than 100 square kilometers in the landscape. These are the most contributing variables, which are distance to protected area and forest loss, which were negatively correlated, and the forest cover and NDVA were positively correlated with the clouded leopard species. In the connectivity analysis, we analyze the least cost paths. It calculates the shortest and most cost efficient route for the movement of animal from one core to another. We identified 59 linkages between 30 cores, of which the LCP between Parking Wildlife Sanctuary and Langton Wildlife Sanctuary was the longest, with a length of 303 kilometers. And the shortest LCP was found between Langton Wildlife Sanctuary and Mullen National Park in Mizoram, with a length of 3 kilometers. We further analyze current centrality for habitat cores and LCP. Current centrality measures the significance of any habitat core or any linkage in maintaining the overall connectivity of the network. High current centrality value means the higher significance. We found that the linkage between Faking and Ulibazhe Wildlife Sanctuary in Nagaland had the highest centrality score, and the Faking Wildlife Sanctuary of Kor had the highest centrality core score. We also
analyzed the cost-weighted distance and Euclidean distance ratio, which is a linkage quality metric. Cost-weighted distance is the total accumulated cost an animal has to bear while moving from one core to another core. And Euclidean distance is the shortest straight line distance between two cores. And low cost-weighted distance and Euclidean distance ratio represents good quality of habitats and linkages because the animal has to spend less cost and less amount of efforts. We found that the linkage between Kale Valley Wildlife Sanctuary and Kane Wildlife Sanctuary in Arunachal Pradesh had the best quality linkage, and a poor quality linkage was found between Dibru-Saikhowa National Park and B.B. Palumani Wildlife Sanctuary in Assam. So, which may be attributed to the high resistance in surrounding landscape. We further analyzed pinch points or bottlenecks for the connectivity in the landscape. Pinch points are those areas within the corridors where the connectivity is compromised and the movement is very constricted and funneled. There are two types of pinch points. This one is adjacent or pairwise pinch points, in which the analysis is performed between the pairs of core habitats, and similarly for each pair of core habitats, this analysis will be performed. This is a more of a local approach with a finer scale, and we found several pinch points in the adjacent pinch point analysis. The second type of pinch point is global or cumulative pinch points, which performs on the landscape level, and it is contrary to the adjacent pinch point. This is a more broader scale approach. And from this map, this shows that the stretch between Namdapha and Intanki National Park had the highest intensity of pinch points. And moreover, however, there is one important point to consider is that we have only considered the Indian part of the distribution range. There is a possibility to for presence of corridors in Myanmar and other adjacent countries. We'll explore that possibility in future in analysis. In conclusion, we identified 30 cores and 59 potential corridors between them. And we can also conclude that pinch points in centrality are vital measures for identifying conservation priority areas. And Brahmaputra River acts as a major natural barrier which separates the northern and southern set of core habitats for carved leopard in India. And Nagaland is an important state to maintain this connectivity. In future, we plan to conduct field surveys and validate these results. And we'll also refine our connectivity model using different distance surfaces. And we'll also include the buffer areas from neighboring countries to make our model more robust and inclusive. I'd like to acknowledge following people. Thank you.
Thank you. Question answer will be in the end of the session. Just for information, there will be a bell at 8 minutes, so don't get spooked and keep looking at the watch over there. I now, Kanishka is going to talk about under the shadow of Big Brother, leopard densities across gradient of tiger densities in Vidarbha landscape. Over to Shikha.
Kanishka has masters from Delhi School of Advanced Studies in Environmental Studies and Resource Management. She joined the institute in 2021 and has been working on this project since then. Over to you, Kanishka.
Thank you for the introduction. Hello everyone, good morning. I'll be presenting on the topic under the shadow of Big Brother, leopard densities across the gradient of tiger densities in Vidarbha landscape. As we know, leopard co-occur with tigers in many places across the Asia, and they have the most widespread distribution among all the felids. And coming to the coexistence, the sympatric species like tiger and leopards, when in the same geographical area, if they coexist together, they do so by implementing various strategies such as spatial segregation, temporal partitioning, differential use and resources, and also composition of predator community plays a role in it. The aim of this study is to investigate the spatial and temporal dynamics of leopard densities through a gradient of tiger densities in the landscape. it postulates that the presence of tiger will have a discernible influence in the spatio-temporal pattern of leopards, while also highlighting the crucial role of habitat covariance. Coming to the study area, so my study area belongs to Vidarbha landscape, and this landscape comprises of 5 tiger reserves, 6 wildlife sanctuaries, and 2 territorial forest divisions. According to previous year long-term monitoring report from the landscape, the minimum number of tigers and leopards was 706 and 390, while the estimates for leopard was 902 and for tiger it was 446 from the landscape. My study area belongs to Eastern Vidarbha. So, for an to optimize results, I've divided the study area into high tiger density area and low tiger density area, choosing Tadubandari Tiger Reserve as high tiger density area and Navigaon Nagsara Tiger Reserve as low tiger density area. So, based on the density estimates from previous year, and both the study sites are located 85 kilometers from each other, and though they are located not so far, but the terrain is slightly different and also the habitat. The terrain of Tadoba is majorly flat with a little hilly area in the southern and northern part, while Nansara have a highly undulating terrain with hilly areas. The bamboo species dominate the forest of Tadoba, while Navigaon Nagsara has mixed deciduous forest. Tiger, leopard, and dhole form the major predatory gill in both the study sites, and wildboar, sambar, nilgai, gaur, barking deer, and cheetal are the principal prey species in both the sites. Coming to the predator and principal prey density in both the sites, which was carried out using camera trap and line transit. So, the prey species, individual density of wild boar was highest in Nansira, followed by cheetal. In Tadoba, the individual density of sambar was highest, followed by cheetal. Coming to the predator density, from last year estimate, there was density estimate of tiger in Nansira was 0.68 per 100 square kilometer, and for Tadoba it was 6.6 per 100 square kilometer. For leopard in Nansira it was 10.63, and for Tadoba it was 9.74 per 100 square kilometer. Coming to the methodology, the data was collected using camera trap, using 2 square kilometer grids in both the study sites, and here are the details of camera trapping. After collecting the data, species sorting and segregation took place using SP-seg, and simultaneously tiger and leopard individual ID was done. We derived covariates. Prey I was also calculated, and other habitat covariates like NDVI, elevation, distance to roads, settlements, and drainage density was derived. To see the spatial overlap, we carried out surface density of leopard and tiger from both the sites, and using SCCR in our program. To see the temporal overlap, we did it for both the predators and with their principal prey, and also using CamTrap-R in our program. Coming to the results of spatial overlap, firstly a little information. So this shows the captures from both the sites for both the predators. As we see, Tadoba has 376 locations where both the predators have captures together, and in low tiger density area it was 76 locations where both the predators have captures together. This was the surface density map generated through SCCR, and then this shows the spatial distribution pattern for both the predators. The value shows the densities, and as Tadoba harbors an optimum density for both leopard and tigers, this shows that there is a very fine level of spatial segregation as we see inside the circles. These are the captures for tigers and leopards. We use quantile regression to obtain estimates of the quantiles of leopard density as a function of tiger density. Given that maximum number of locations had tiger and leopard captures together, we see a positive association between the density of. Coming to the low tiger density area, here because tiger density was so low, we see a very distinct level of spatial segregation in the spatial distribution of both the predators. These are the captures for tigers and leopards. Here, as the number of tiger and leopard captures together was so low, and also leopard have outnumbered the tigers at many locations. Initially, at lower contact, we see a slight incline in the slope, but there comes a point where leopard density is not really affected by the tiger density. Hence, we see a saturation in the slope. Coming to the results of temporal overlap. This shows the activity overlap between predators, leopard and tiger, from both the sides. It was 92% and 88%. And though there is high activity overlap, but we see a fine adjustment in the activity peaks. And with the principal prey, in Cordoba, tiger highly overlapped with boar and leopard highly overlapped with sambar. And in Navegaon Nagsera, tiger highly overlapped with wild boar and leopard highly overlapped with sambar. It was likely that leopard could potentially avoid tigers at a very finer level, finer temporal scale. So we did a preliminary study by taking a sample of our data to see the pattern, to evaluate that pattern, and we saw the lag between photo captures of leopard after a tiger has been photographed. So here the minimum lag varied from 28 minutes to 0.8 days, which is 19 hours, and the maximum lag varied from 6.7 days in low tiger density area and 33.8 days in high tiger density area. Coming to the conclusion, by seeing the spatial temporal pattern, and also it can be said that interaction between both the predators can vary from site to site depending on various factors such as density of the predators in that site, the distribution of prey, and also the habitat of that area. And according to our hypothesis, we hypothesize that if there is a high overlap in one niche component, there will be a low overlap in another. So as we saw in high tiger density area, because the spatial segregation was so low, the temporal segregation was high. And in low tiger density area, we saw the spatial segregation was so high, so the temporal segregation was low. And as you know, Nagsera is now involved in tiger translocation program. This study will give insights into the interplay between the predators inside the reserve. As way forward, I'd like to say that there should be more studies on radio telemetry for both the predators where they coexist together in high densities for say Tadoba, and which can give more information and elucidate their avoidance mechanism at a finer scale, and by providing information such as kill rates and prey handling, which can be essential in understanding their interference interactions. I'd like to acknowledge following people. Thank you.
Thank you, Kanishka. Now I invite Anubhuti Krishna. She is going to talk about individuality methods, learning from tiger translocation experiments. Over to the Krishna.
Anubhuti has a master's in wildlife science from Wildlife Institute of India in 2019-2021 batch. She joined Wildlife Institute of India as a researcher in the long-term monitoring of tigers and co-predators and prey in Vidarbha landscape in 2022. Research interests include carnivore ecology, their community process and movement, along with spatial ecology. Over to you.
Thank you, Dr. Shikha. A very good morning to one and all present here, and thank you for being here this morning. Today I'll be talking about our learnings from the tiger translocation experiment, which has taken place in Navegaon Nagsera Tiger Reserve, and how individuality matters and should be taken into consideration when such conservation measures are taken. So Navegaon Nagsera Tiger Reserve is located in the northeasternmost portion of the eastern Vidarbha landscape of eastern Maharashtra, and it plays a pivotal role as it acts as a stepping stone corridor within this landscape. It connects the various protected areas within the landscape and also with protected areas further onwards in Madhya Pradesh, Chhattisgarh and Telangana. So NNTR over the years has faced what I like to call tiger troubles. So the total number of tigers within NNTR has remained more or less consistent and similar across the years, even though there have been several cubs which have been born during this time frame. And we attribute this majorly to a male bias sex ratio. So the ideal male to female sex ratio for tigers is 1:3, and we can clearly see that the sex ratio over the years has been male biased in NNTR, contributing to the steady number of tigers and no increase in population. A simultaneous study also estimated the carrying capacity of tigers within NNTR, and it was estimated at 20 adult tigers for the tiger reserve. With this backdrop in mind, a conservation translocation program was proposed for the tiger reserve. So what exactly is conservation translocation? It is a deliberate movement of organisms from one site to another where the primary objective is a conservation benefit. And these fall within two categories, either reintroductions or reinforcements. And since NNTR already had a pre-existing tiger population, this falls within the category of reinforcement. So for the first phase of the conservation translocation, two females were captured, one from Brahmapuri Forest Division and one from Talubandhari Tiger Reserve, collared, and then subsequently released on the 20th of May 2023 within NNTR. So before we look at what happened post-translocation, let's first meet the two tigresses. So NT1 was captured from Brahmapuri Forest Division, which is a human-dominated landscape, and was about 30 months old at the time of capture, whereas NT2 was captured from Talubandhari Tiger Reserve and was 22 to 24 months in age at the time of capture. So with this backdrop, we will look into how these two individuals behave differently and how individuality played a role post-translocation. So here we have a map representing the movement paths obtained from the GPS collars of the two individuals post-translocation. So NT1, the individual captured from Brahmapuri, has moved from NNTR, which is shown by the blue path here, has moved from NNTR all the way across the Maharashtra-MP border and has actually dispersed to the patches of the Balaghat Forest Division in MP. Whereas NT2, captured from Talubandhari Tiger Reserve, shown by the yellow path here, has stayed within the bounds of the Tiger Reserve. So if you look at this movement path here, NT1, right after release, had already moved towards the peripheral portion of the Tiger Reserve, close to the buffer boundary, and was restricted to that particular area because of the summer months and availability of water only within those restricted portions. And then with the onset of the monsoon, we suddenly saw an increase in the foraying movement and has dispersed across the agricultural matrix into the forest patches of the Balaghat Forest Division, and has been there since the end of July and has tried making forays within the forest patches but has remained restricted more or less to that region. Now if we look at the second individual, NT2, right after release, she made several foraying movements within the Tiger Reserve to explore this new area, but then just like NT1, was situated near the boundary of the Tiger Reserve, restricted by the availability of water during the summer months in the form of various water holes here. And then as soon as the monsoon arrived, this individual also showed increased movement across the landscape, but within the Tiger Reserve itself, and has been exploring this area since. So now looking at various movement metrics and parameters for these two individuals. So NT1, the individual which has dispersed from the Tiger Reserve, travelled a total distance of 737 kilometers over the 3 months of data that has been collected from the collar, whereas NT2 has only covered a total distance of 421 kilometers and an overall displacement of 63 kilometers from the release site to the last location has been observed for NT1, whereas NT2 is only about 6 kilometers away from the release site, with the two release sites being very close to each other, only 4 kilometers apart. Now looking at step length and step speed, so if we assume these orange circles to be representing consecutive GPS fixes obtained from the collars, so distance between any two consecutive points represents a step length. So the step length for NT1 again showed a higher mean value than that for NT2, and the mean step speed for NT1 was also higher than that for NT2. So NT1, the individual which dispersed, travelled farther distances between two consecutive GPS fixes and also did this more quickly. Now looking at a different movement metric, which is directionality. So again, if we look at the illustration, if an individual is moving from point A to point B to successive GPS fixes, then the dashed or dotted line here represents the trajectory we would expect that individual to take. So now considering that the individual moved to point C and changed their direction of movement, then the angle marked here represents the turning angle for this individual. And a mean turning angle of 0 hence represents directed movement, whereas a mean turning angle of pi or minus pi represents undirected movement. So looking at the two individuals, for NT1, the individual that dispersed across the landscape, and the points have also been divided into day and night GPS fixes. So for the day parameters and day locations, there was no directionality or clear directed movement for the NT1 individual, as the red arrow is closer, the mean angles are closer to the value of pi. Whereas at night, we could clearly see directionality in terms of the movement taken up by NT1. Conversely, for NT2, values for both the means, the day as well as the night, were close to pi, and there was no clear directionality in movement either during the day or night, since the individual has moved more or less in circular paths within the tiger reserve. Having a look at daily distance moved, so the blue line here represents the dispersed individual, whereas the orange line represents the resident individual. So again, NT1 showed greater values of mean distance, daily distance moved, so distance moved across a 24-hour time window as opposed to NT2. A similar trend was observed for daily displacement. So daily displacement would be the distance between the first and last locations of a 24-hour time window. And a similar trend was also seen for average daily speeds. We also observed that seasons play an important role in the movement metrics estimated, since we observed that the two individuals were restricted to certain areas during the summer months. So during the summer months, both NT1 and NT2 showed similar values for distance moved in kilometers per day. However, with the onset of monsoon, we can see that the value for NT1 increased as it dispersed across the matrix of agricultural fields pretty quickly to the forest divisions of Balaghat. Whereas for individual NT2, the movement parameters actually decreased during the monsoon months, since it had already more or less established a territory within the tiger reserve. A similar trend was observed for displacement, where again the summer values were similar for the two individuals, whereas for NT1 the value increased during the monsoons and decreased for NT2. The plots for average daily speed also show a similar trend. Now having a look at space use, so there are different ways to understand space use. So with the example of with the help of an example data set, the first way of looking at space use is the minimum convex polygon, which basically looks at the geographical spread of points and only looks at geographical distribution. An improvement on this is kernel density estimation, which looks at clustering of points and then draws up contours for different percentage values. And then this is further advanced by the Brownian Ridge Movement Model, or BBMM, which incorporates a time parameter into the KDE model. So it looks at time spent at these clusters and then draws up a distribution. So for the two individuals, since they are both dispersing individuals and they carry their home range with them as they move across the landscape, so MCP and KDE did not provide very accurate representations of the space use. And hence the BBMM model was used to estimate space use for these two individuals, again with individual NT1 showing greater values for the space utilized, the utilization distribution, as opposed to NT2, since it has covered a larger scale of landscape. Now looking at some of the conservation implications and outcomes of the study and the learnings from this whole translocation experiment. So different individuals show varying patterns of movement post-translocation. And if we revisit our two individuals, then we realize that yes, individuality does matter. So individual NT1, which was captured from Brahmapuri Forest Division and was about 30 months old, had just recently established a territory within Brahmapuri Forest Division when it was captured for translocation. And this, we believe, may have played a role in greater homing instinct and hence directed movement from NNTR to Balaghat Forest Division. Whereas NT2, which was only about 22 to 24 months old, was actually in the dispersal state, as the translocation acted as an aided dispersal movement. Several translocation programs are now being taken up for conservation of tigers and are gaining more and more importance. So it is important to choose wisely which individuals we translocate and where. And additionally, NT1 also elucidated a functional corridor between NNTR and Balaghat Forest Division. And stepping stone habitats such as these small forest patches within a matrix of agricultural fields play a key role in highly fragmented central Indian landscape dispersal of large carnivores. I would like to thank the following people. Thank you.
Thank you, Anuruti. And now I invite A. Krishnan. He'll be talking about cost of prime reality, survival of tiger cubs under the influence of high turnover and density.
Krishnan has done his bachelor's in botany from University of Delhi in 2015. After his master's in environmental science from Rajasthan, he joined Wildlife Institute of India as an intern working on Radiolium Bustard project in 2018, and later is joining the current project, about which he will be presenting now. Over.
Thank you, Dr. Shikha. My name is A. Krishnan, and I'm working in the long-term monitoring project in the Leopard landscape. Today I'm going to present the topic cost of prime reality, which basically deals with the survival of tiger cubs in high tiger density and high male turnover areas. So first we will start with a little bit of tiger ecology. Tigers are generally solitary and nocturnal in nature, and they come together only during mating. And the mating takes place around for 2 to 7 days, and after which the gestation period is of around 102 days. Then the female gives birth to cubs, which is and the litter size is around 1 to 7. The female takes care of the cubs. The males rarely help in that. The cubs stay with their mothers for around 20 to 28 months, after which they disperse and establish their own territories. Now, when we consider breeding, for a female, bringing up the cubs to adulthood can be considered as breeding success. And giving birth to cubs and taking care of them is a large investment because it takes most of the time and effort of the female to take care of them. Now, next thing is the threat to cub survival. Cubs are very vulnerable and can easily be killed. Mostly, they are killed by co-predators as well as rival males who are not their fathers, so that they can mate with the females. Now, coming to the study area, my study area is Sadovandari Tiger Reserve, which is situated in the eastern part of Vidarbha, in the Chandrapur district. The vegetation can be broadly categorized as southern tropical dry deciduous, and it is dominated by bamboo. Inside Tadwa, I am focusing on the core area, the data has been collected from the long-term phase 4 monitoring and occasional field observations from 2014 to 2023. So the study area is abundant prey base as well as plenty of perennial water resources and mix of habitat, which is ideal for the tigers. In here, we monitored 2 tigresses, T7 and T12, to look at their cub survival rate. Now, coming to tiger population in Tadwa, the population of tigers in Tadwa has remained around 85 since the year 2015, and the sex ratio has been around 1.25 since 2019. Which has led to higher competition between males for territories and mating rights. As can be seen in these maps, there is large overlap between home ranges of male tigers in Tadwa in the prime areas.
Now coming to males. Now males can hold their territories for a few years in their primes, around 2 to 4 years, depending on their abilities and other factors. In this case, it is a map showing the movement of territory of male tiger T33 Wagdo. Wagdo is the oldest recorded male tiger to live in wild. He lived for 17 years in the wild, and he died in last year in 2022. Initially, he established his territory in Kor and stayed there till the year 2014. After which he moved to, he was pushed by younger males towards the buffer area, Morley buffer. And then later on, there also younger males came in and pushed him towards the southern part of the tiger reserve. Again, after that, even more younger males came and pushed him towards the territorial forest in the southern part. Now, when we look at Wagdo's tenure, he stayed in the core area Morley from 2010 to 2014, and during which time he had 3 litters with 3 separate females, and a total of 10 cubs were born from this. And then he was pushed by the male T44, and he went to Morley buffer, where he stayed for 2 years and mated with 1 female and gave birth to 1 litter, which had 4 cubs. Then he was pushed again towards the boundary of Morley buffer and Chandrapur buffer by the male T50. Then in that area he stayed for 2 years again, and then he had cubs with 2 different females, of which 7 cubs were born. Again, from there also T46 tiger pushed him towards the territorial forest. Now coming on to the females that we have monitored. This is the first female, T7, Choti Tara. Now Choti Tara has established a territory inside the core area of Tadaba from 2014 to 2023. She hasn't changed her area. During this tenure, during this time, she has given birth to 5 litters, of which a total of 12 cubs were born. And as can be seen, there have been multiple males in our territory which have affected the cub survival rate. Similar to Tara, the next female we are looking at is T12 Maya. Now Maya is the most famous tiger of Tadaba and the major attraction for tourists. Tourists come in large numbers to see her and her cubs. Maya, again, she also has established a territory inside the core from the year 2014 to 2023. Her area has also not changed much. There are plenty of water bodies and meadows in this area. Yeah, Maya also has given birth to 5 litters, out of which 13 cubs were born. But unfortunately for Maya, only 2 cubs have been able to survive till adulthood. And this can be attributed to the presence of large number of males and high turnover rate of males. As can be seen, Maya has given birth to 5 cubs from 5 different males, as compared to Tara, who has given birth to 5 cubs from 3 males. The effect of low cub survival the low cub survival rate can affect the population dynamics of the area. And if the survival rate of cubs is low, then the population will be declining. Now, strategies adopted by the tigers in Tadaba. First strategy is delayed dispersal of subadults, and the second strategy is mating of females with multiple males. Now, looking at the first strategy. In this case, on the right side, we have T49, Matkasur, and next to him is his son, T126, Chota Matka. Now, Chota Matka stayed with his parents for 1 more year after reaching adulthood, and this helped T49 Matkasur to extend his tenure from 3 years to 4 years in the presence of rival males. Similarly, in the case of towards the right, we have T171 Bhima. Bhima stayed with his parents again after reaching adulthood, and his parents gave birth to next litter. Now Bhima took care of these cubs when his parents went for hunting or patrolling, and this helped in increasing the survival probability of these cubs. So the next one is faking with multiple males. This strategy has been seen in Tadaba by multiple females, and In this case, it is T12 Maya, which we have seen, and she has mated with multiple males within a span of few days. And what does this is all these males, the cubs are born. They believe that the cubs are theirs, and so they don't kill those cubs. Now coming to yeah. So finally, only 2 cubs out of 13 born to Maya and 5 cubs out of 12 born to Tara have been survived, which brings a total to around 7 cubs out of a total of 25. This leads to around 28% of survival probability of these cubs. High density and turnover of males in the study area seems to affect the negatively impact the cub survival rate. And also, if the connectivity is hindered, the male dispersal will be reduced, and which will lead to high competition between these males, and hence low cub survival probability. Again, cub survival probability will negatively affect the population and will help reduce will lead to declining population and in the study area as well as the larger landscape. Due to limited sample size, statistically significant inferences have not been drawn, and a larger sample size from long-term study is required for this, and information associated with the other covariates is also required. I would like to acknowledge the people, and finally, I will leave you with this video, the most viewed tiger video in the recent time. So the cup in this video is T12 Maya's last litter, and the sad reality is that even this cup was not able to survive in these harsh conditions.
I now invite witness what I read.
Ankit is from Maharashtra and he's done his master's in wildlife conservation action from Bharati University. He's worked on forestry and central Indian landscape, securing and securing wildlife corridors. He joined this project in 2022. Over to you.
Thank you, ma'am. Very good morning, everyone. I'm Ankit, and I'll be talking about the readiness for tiger recovery in Buxa Tiger Reserve, which is a situation in business. And this is basically an assessment of the work we have done in the phase 1 for the project, which started from 2018 to 2023. In the past, tigers have existed in Buxa in good numbers, and this has been cited in multiple literatures as well. With the onset of anthropogenic disturbances, we somehow get extinct. And coming on to the objectives of our tiger recovery project, we first focused on strengthening the monitoring protocols for the tiger reserve, along with capacity building of the frontline staff, mapping all the geospatial layers, several administrative as well as ecological parameters. Annual prey-based assessment, monitoring of tigers, and eventually develop a landscape plan for tiger movement in the context of tiger movement. We'll be assessing the status of tiger recovery broadly over 3 topics: status of habitat, status of prey, status of tiger, and co-predators. So, starting with the status of habitat, it's predominantly a dry evergreen forest with dense undergrowth. We started compiling several spatial layers and to generate several maps, for example, forest cover map, NDVI map, location grading, and so on. In order to study the habitat suitability of tigers in Buxa, we performed a multi-criteria analysis on the basis of the mentioned layers, and all the layers were first reclassified and assigned a value between 1 to 9. Then the reclassified raster was multiplied by the weightage given to each layer, and all the layers were added to get the final output. All this was done using a raster calculation, raster calculator. And coming to the results of the same, we can roughly say about 40% of the area of tiger reserve is either highly or moderately suitable for the tigers. The tiger reserve also faces a significant amount of anthropogenic disturbances, having 37 forest villages and 4 major FD holdings within the tiger reserve. So the inevitable pressure on forest resources and associated illicit activities. These are the village locations of all the villages that are coming inside the tiger reserve boundary. Based on the capture rate data from the camera traps, a bubble map was generated to demonstrate hotspots of human and livestock activity in the tiger reserve. So overall, the habitat characteristics indicate a dense forest cover with a varied level of human disturbances. Moving on to the status of prey, we analyzed 2 data sets. First, the herbivore estimation exercise, which is the phase 1, along with a systematic camera trapping exercise that we have been doing. So on the basis of this, the blind transit results, barking deer showed the highest density estimate of 6.38 per square individuals per square kilometer with a standard error of 0.85. And the overall ungulate density in the reserve is estimated to be 12.17. The same data has been analyzed and a breed-wise encounter rate map was prepared to understand the distribution of prey species throughout the tiger reserve. Coming to the relative abundance values of based on the camera trap data, it is clearly the barking deer having the highest capture rates, the RA values. And the same data was interpreted in preparing the species occurrence map. So it's quite visible, barking deer along with the wild pig has been the most widespread species throughout the tiger reserve. So overall, prey density in the reserve appears to be steady. With regular prey augmentation and habitat management, the prey population can be increased. Moving to the status of tigers, currently there are no tigers in Buxa Tiger Reserve. And with the continued efforts of our tiger recovery project, we were successfully able to photo capture a tiger in Buxa, and this is in fact the first photographic record of a tiger from Buxa. These are some locations where this individual was captured. Moving on to the status of co-predators, it has a thriving diversity of co-predators, and leopard showing the highest value of RAI based on the camera trap data, followed by leopard cat, dhole, jungle cat, clouded leopard, golden cat, marble cat. Similarly, we made a species occurrence map, and common leopard along with leopard cat is showing the most widely distributed co-predator in the tiger reserve. And the clouded leopard, golden cat, marble cat can be very clearly seen restricted to the northern hilly parts of the tiger reserve. So a regular capacity building program has been our key component. This has been the major activity, one of our major activity and quite focused one. And starting from 2018, in complete phase 1 of our project, we have completed training of about 124 individual training programs with a total gathering of 2,742 individuals being trained. We also happened to radio collar leopard 2.5 years back, and monitoring this leopard also helped us in imparting hands-on training to the forest staff about radio telemetry, the equipments used, etc. And this may be helpful in the future if there's a tiger reintroduction program in the reserve. Community engagement through a detailed questionnaire survey to understand people's perception towards tiger reintroduction and assessing their socio-economic status has been initiated. It's an ongoing activity. Survey in 2 out of 6 of our target villages have been completed. Moving on to the discussion part, Uh, we'll be discussing about the habitat improvement work that we are doing and will be continuing in the phase 2. Secondly, the carrying capacity of tigers and the connectivity. In habitat improvement, we have identified potential grasslands that can be developed as a part of habitat improvement, and also identification of areas with hotspots of animal movement to prioritize protection measures. This were done. And habitat identification, the grassland identification was done, mapping the invasive species distribution in the tiger reserve. This was based on the vegetation sampling data. This later carrying capacity of tigers. In order to study this, we first assess the ungulate density of the tiger reserve, as I mentioned before, and also based on the directives of NTCA in 2022 last year, a microcore within the core of the tiger reserve has been identified. So microcore is basically the most inviolate area of the tiger reserve in the context of human disturbances. And also, I just forgot, the ungulate density in the microcore has been found to be 33.6. So carrying capacity, we calculated the carrying capacity in two sets. First, based on the prey and preferred prey density. Secondly, based on the predictive equation with preferred prey biomass. So this first result is based on the prey density with the predicted abundance of tiger coming to around 10. The same result after analyzing with the preferred prey density comes down to the predicted abundance of tiger to 4.86. And the next part, understanding the carrying capacity on the basis of predictive equation of Hayward et al. Based on the prey biomass, the predicted tiger abundance came to 37. Speaking about the connectivity in brief, we have prepared a landscape map of the protected areas, and also the red dots in the map are the tiger locations of the same individuals that we have photo captured in 2021. So this briefly shows how the tiger actually entered Buxa Tiger Reserve from the east. It stayed there for a while, moved, was recorded in Leopard National Park, moved to and further landing up at Mahananda Wildlife Sanctuary. So the last record of the photographic record of tiger was found at Mahananda Wildlife Sanctuary. We have matched the stripes. It is the same individual which was captured in Buxa, and it has gone resident now in Mahananda. In the end, we can sum it up as the continuous prey augmentation along with the existing wild ungulate density and effective protection by trained staff. Tiger recovery in Buxa is possible through natural colonization and conservation assisted translocation. So in the end, I would like to acknowledge all the foreign people for their efforts. Thank you so much.
Thank you, Ankit. Now I invite Jaydeep Patil. He is going to talk about the correction of large carnivores in Leopard.
Jaydeep is from Kolhapur, Maharashtra, and has completed his post-graduation in botany in 2014. He's worked with various NGOs monitoring large carnivore populations in conflict-prone areas of Vidarbha. And since 2019, he's been working in this current project. Over to you, Jaydeep.
Thank you. Good morning, everyone. I'll be your presenter for this session, and I'll be talking about the interaction of large carnivores in Leopard Tiger Reserve, Maharashtra. Coming to the project details, this study has been a part of the long-term monitoring of tigers, co-predators and prey in tiger reserves and other tiger-bearing areas of Vidarbha, which is a project funded by the Maharashtra Forest Department for the duration of 10 years. We aim to understand the patterns of co-occurrence between three sympatric large carnivores, that is, tigers, leopards, and does in Vidarbha landscape of Maharashtra. And as for the methodology, we used camera trap data that we collected in the years 2020, 2021, and 22 during the phase 4 exercises conducted in the study site. This data was further segregated using AI tools like Megadetector and SPSeg, that is Species Segregator. Softwares like Timelapse, R, and ArcGIS Pro aided in the further processing and analysis of the data. Coming to a study site, on the eastern front of Vidarbha, nestled in the Satpura Hill Ranges, lies Melghat, fourth largest in the country and largest in Maharashtra. Melghat Tiger Reserve covers an area of nearly 2,800 square kilometers with a core of 1,500 square kilometers. It is one of the first 9 tiger reserves that were formed in 1973-74. The wide variety of flora, fauna, and even habitats that Melghat hosts makes it truly magical. From the lofty peaks of Vairat to the valleys of Belkund and from the serpentine rivers that make their way towards the river Tapi to the meadows left in the wake of the translocated villages, Melghat has it all. It also hosts the endangered forest owlet along with a trove of other avifauna. In herbivores, Melghat hosts a number of species like gaur, sambar, barking deer, spotted deer, nilgai, four-horned antelope, wild boars, chinkara, blackbuck, and even langus and rhesus macaque are found in large numbers in the reserve. Coming to the carnivores, Melghat at last count in 2022 held 55 tigers and 168 leopards. Dholes have also been observed in smaller numbers, and their pack size is also observed to be small in the reserve. Next, we come to our results. We studied the temporal activity of our focal species and understood that in 2020, tigers and leopards show irregular temporal activity throughout the night hours. Dholes, on the other hand, displayed a strong unimodal activity with a peak at 6 a.m. in the morning. Due to lower number of tigers inhabiting the eastern region of Melghat, we may be seeing the higher spatial overlap with leopards and dholes in this region. In 2021, we see a change in dhole activity with the peak shifting from 6 a.m. to 7 a.m. and tiger and leopard activity shows a higher temporal overlap in 2021, which can be attributed to their space use patterns. They show a higher degree of spatial segregation, which may be allowing them to co-occur in the same temporal frame. In 2022, we see a marked change in the dhole activity patterns with their peaks changing from unimodal to bimodal with peaks at 7 a.m. and 5 p.m. While tiger and leopard activity periods seem to have a higher degree of overlap. Those space use patterns appear to be further segregated from tigers and leopards in 2022. This may explain the shift from unimodal to bimodal temporal activity patterns. Here's a comparison of the temporal activity patterns across the years for all 3 focal species, and similarly the comparison of their spatial distribution across the years in Melghat Tiger Reserve. Coming to the percentage of photo captures across the years, we see a higher percentage of camera traps that captured both tiger and leopards as compared to those that captured both leopards and dholes. Whereas tigers and dholes show the least percentage of captures in the same camera trap. In 2021, the percentage of camera traps that captured both tigers and leopards as well as tigers and dholes has increased markedly. Whereas in 2022, we see a pattern emerging similar to the previous years where tigers and leopards were captured in higher percentage of cameras followed by leopards and dholes, and tigers and dholes have the least percentage of captures across the camera traps. Tigers, being the apex predator, have shown little change in their temporal activity throughout the years,
And their spatial distribution also indicates that the hotspots are mostly situated around higher prey numbers and prime undisturbed habitats. Leopards, on the other hand, have shown a marked increase in their temporal activity towards 2022. Space use of leopards throughout the years also shows a changing pattern, which can be either in avoidance to its co-predators, or it may be attributed to prey selection. Dholes have shown a clear change in their temporal activity patterns, with a new activity peak emerging towards 2022 at 7 p.m. in the evening. Dholes may be unable to compete with tigers or leopards, or may prefer different prey species to tigers and leopards. This may shed light into their spatial patterns and requires further investigation. In conclusion, we can say that tigers are avoiding daylight hours but are active throughout the night. Leopards show changing spatial patterns over the years to possibly avoid territorial overlap with tigers or due to prey selection. Those display both temporal and spatial avoidance to tigers and leopards in some cases. I'd like to thank the following people, and with that, I conclude this session. Thank you.
Thank you, Jaydeep. And now I invite Asuraj Lage. He is going to talk about silent strides, mapping carnivores and ungulate distribution in semi-arid regions of Karnataka.
Basavaraj has completed his integrated BSc MSc in Biological Science from Bangalore University 2018. Has been associated with Wildlife Institute of India since 2018, currently working in the project Ecology and Conservation of Carnivores and Ungulates in Semi-Arid Landscape of Karnataka. The project focused towards generating a baseline data for faunal species of semi-arid landscape. Over to you, Basava. I also request everyone in the audience to pass on their questions. We'll be taking questions next, and the online audience to write their questions in the chat box. Thank you.
Good morning, everyone. I'm Basavaraj Mulle. I'm currently working as a project fellow in a project titled Ecology and Conservation of Major Carnivores and Ungulates of Semi-Arid Landscape of Karnataka, under the guidance of Professor Kamal Qureshi, Dr. Vishnupriya Kulipakam, and Dr. Leopard. So the project is funded by Karnataka Forest Department, and the project duration is 2021 to 2024. So today my talk is titled as Silent Strides, Mapping Carnivore and Ungulate Distribution in Semi-Arid Regions of Karnataka. So let's talk about why this study was so important. So in India, conservation initiatives are always centered around protected areas and their inhabitants. Karnataka is well known for tigers and the elephant population. Having said that, the semi-arid landscape also provides a crucial habitats to some of the endangered species like Indian grey wolf and great Indian bustard, chinkara, and black buck. So through this project, we are trying to address some of the questions such as where these animals are present, what is the distribution pattern, and what is the density of this population. Not only density, we are trying to assess the population parameters through which we'll be generating the baseline data for the entire landscape. This brings me to the study area. Our study area spans across 10 districts of eastern plains of Karnataka, which consists of 67,000 square kilometers. So currently we are working in a Ballari circle, which has 5 forest divisions, and currently we are working in Koppal, Ballari, and Vijayanagara districts. So these are some of the key habitat types. I'll quickly walk you through the heterogeneity of the landscape, so you will get to know the landscape features. So our study areas majority consist of farmlands, interspersed with the rocky outcrops and scrublands, and followed by mining areas. So to address these questions, we followed double sampling approach, in which the first phase of the methodology says that in the first phase of the study, we conduct large-scale surveys across the study area using interview-based surveys in an occupancy framework. So we overlaid entire study area with the 25 square kilometer grids, and in each grid we conducted 5 interviews, and those 5 interviews are spread across the grid so that we cover every corner of the grid, and the distance between the interviews were 1 kilometer. We collected data on presence and absence of the animal and also the subsidiary data of the species. Having said that, we use single season occupancy model in R and analyze the data. So these were some of the covariates that was used in modeling the occupancy. So I just want to emphasize on the sampling design of the study. We adopted adaptive cluster sampling, in which it's a multi-level sampling where you select the random grids initially, and once those random selected grids are they record any presence of animal, then we end up surveying the surrounding grids also. So this is the kind of information we collected while we interviewed people across the landscape, and this is the pictorial representation of the interview locations across the landscape, which is of 15,000 square kilometers, and we conducted around 2,682 interviews. This is a demographic variation among respondents. We were able to capture demographic variables such as age class, education classes, professional classes, and sex classes. So coming to the study species, Indian grey wolf. The Indian grey wolf distribution was, the proportion of area occupied was estimated to be 0.53, and the distribution is, Indian grey wolf was found to be widely present across the study area, but more dense towards the eastern part of the study area, mainly due to one plausible explanation for this distribution pattern observed was the terrain, the flat terrain, which also hosts black buck. So wolves are known to follow the black bucks. So the following plots are based on the perception of our respondents. Our respondents reported that they encounter wolf on a monthly basis during monsoon time and during late evening hours, and these animals were seen in man-made landscapes such as farmlands, and their diet was mainly consist of livestock. Our respondents also reported livestock damage as one of the conflict types, and these events were observed on a monthly basis during late evening hours, and this was highest recorded in monsoon, and again these events were observed on man-made landscapes such as croplands in which livestock are raised, and then the mitigation measures used against to mitigate the conflict was guarding, in which you use guard dogs to protect your livestock. Coming to the second study species, Bengal fox. The Bengal fox distribution is seen on the southern portion of the study area, and the proportion of area was occupied was estimated to be the 0.74. The one possible explanation for this distribution pattern is due to the heterogeneity of the crop being grown in that landscape, where understandably the Bengal fox diet mainly consists of crops in the study area. So our respondents reported that they encountered fox on a daily basis, as during monsoon. Again, the animal was seen on evening hours and man-made landscape, understandably, because man-made landscapes such as farmlands in which they are feeding on crops. Our respondents also reported that the crop depredation as one of the conflict types, and it was observed on daily basis, late evening hours, monsoon, and guarding was one of the mitigation measures adopted. Coming to the golden jackal distribution, the golden jackal distribution is restricted to the western part of the study area. One possible explanation is due to the presence of highest number of scrub forests in that area, and also the crop pattern. So this is a perception where the respondents recorded the animal on a daily basis during monsoon, late evening hours, man-made landscapes such as farmlands, and animal was reportedly feeding on crops. Our respondents also reported human jackal conflict. Crop depredation was one of the main types, and it was again recorded on a daily basis during monsoon, late evening hours, and guarding was the method used to mitigate the conflict. So coming to the next study species, common leopard, Indian leopard. The proportion of area occupied was estimated to be 0.29, and leopard has a patchy distribution. It's only distributed across in the central part of the study area. Understandably, because one plausible explanation for this landscape is because of the presence of refuge sites such as rocky outcrops, which are the typical habitats of the landscape. Our respondents reported that the animal was seen on a monthly basis, and again, animal was encountered it was highest during monsoon, late evening hours. The animal was encountered in natural landscapes such as scrub forest and rocky outcrop, and the animal diet was consist of livestock as reported to the respondents. And respondents also reported that livestock damage is one of the main conflict types. It was observed on a monthly basis during monsoon and late evening hours. The conflict was encountered, or habitat of conflict was reported to be man-made, such as farmlands and natural landscapes, and the guarding was the method used to mitigate the conflict. So coming to the next study species, striped hana, which also has a similar pattern like in a common leopard. Understandably, the distribution is restricted to the refuge sites, which are rocky outcrops, and the proportion of area occupied was 0.21. And this is a perception recorded to the thing. I'll just quickly walk you through the slides. So coming to the next study species, sloth bear. So sloth bear again is distributed in the central part of the study area due to the presence of rocky outcrops, which are refuge sites for these animals, which are the last remaining refuge sites for the animals. And these are the perception. The animal was encountered on a daily basis during monsoon, late evening hours. Animals in man-made and landscapes such as agricultural crops, agricultural lands, and it was recorded to feed on crops. And the conflict was also reported. Crop depredation was one of the major types recorded on a daily basis during monsoon, and man-made landscapes such as farmlands was the site where conflict happened, and guarding was the method they used. So coming to the last study species, black buck. Black buck distribution is a patchy distribution restricted to the last western part of the study size, and one of the possible explanation is that the terrain again, as you saw in the Indian grey wolf distribution, which also has the similar sort of distribution, and these were the perception reported by our respondents. And so we had less number of respondents who reported pangolin presence. So nevertheless, we had some presence. So we could not model this. We only fitted the model using the interceptor model. So these are the perception associated with the species. The conclusion way forward, this study will enable us to identify the wildlife hotspots in which we will select the intensive study sites and we deploy camera traps and assess the population parameters, such as population size, and we also aim to follow individuals and study the ranging pattern, behavior, and so on. So I would like to approach the following people. Thank you.
Thank you, Basuraj. I invite other speakers on the stage. Also, you can stay there. Basuraj, you can stay there. Others can stay there. We are going to ask Dr. Salvador will be asking questions. I need technical help. Somebody please put on this projector. Is projector on? Come on this side. Online questions are being sent. So we are going to reduce the questions depending on if some of you are getting more questions, and those questions can be asked during tea time. Over to Dr. Salvador.
Thank you, sir. It was wonderful listening to all the talks. Now it's time for questions. The first question, Tribhuvan is here, and there are two sets of questions. Tribhuvan, this first question is from Oyavan. I don't know if you can focus this, but I'll read it out. What was the criteria for selecting the course? The entire landscape is forested. Have you considered the night light intensity while selecting the core? Right? That is the first question. And you can answer the first question and we go to the second one.
Thank you, Anne, for the question. We selected the core habitats on the basis of the high suitability criteria and which had an area of more than 100 square kilometer, which we consulted with other experts, and it was the minimum area which can sustain a small population of clouded leopards. So that's how we selected the core areas from high suitability patches.
Okay, probably the reason why he's asking is because cloud leopards are known to be in forested areas, not those free areas, but outside free areas also.
In our analysis, we also found the core areas outside the PAs also.
So the second part is NULC has built up area which is an indicator of human habitation. We also use human habitation separate. Please.
We did not use built-up area as separate covariates. We used built-up area for to analyze the connectivity and the weightage to assign the resistance score for the connectivity to construct the resistance surface.
Okay, moving on to the next set of questions for this question by Shikha. Given the short dispersal distances of cloud leopards, do you think the corridors identified by your study will be used or are being used?
So, according to the previous studies by McDonald in Thailand, it was observed that the maximum mean dispersal length of for cloud leopard was 252, around 150 to 252 kilometers. That's how we put a threshold on the length of corridor, and we did not consider any corridor above 300 kilometers. And we will verify these corridors or use of these corridors once we do the field studies.
The second part is, what about connectivity as a covariate in your model, since cloud leopards are which is the ecology of cloud leopard is mostly associated with tropical forests.
We included forest cover as a covariate for STM, which was also positively correlated with the presence predictability of cloud leopard, and it is considered that it is a highly arboreal species. But in recent research, we did not find any other authors did not find any proof that it is highly arboreal. It is arboreal, but not as much as previously thought.
Thank you, Dravid.
Sure.
But I wanted to ask you a question. Is it going to be a practical feasibility of assuming connectivity of all PS in the landscape you are working? Because it is going to be a practical solution of identifying clusters of protected areas. We have a whole range of protected areas. I'm asking this question because the landscape and the ownership of the forest in the northeastern India is practically different from the rest of the India. So I think, are you going to suggest connectivity as the factor or a cluster of protected area as a possible solution to the issue?
So we have just analyzed the functional connectivity as in on the modeling and mapping basis. So we are going to verify these corridors or the usability of these corridors once we do the field study. Yes. So we'll fine tune the model once we collect the pre-data.
Okay, thank you. Thank you.
So we limit the questions to 2 questions per speaker.
2 or 3 maximum.
We are running out of
Kanishka.
time. Kanishka, your question is, sir, would you like to ask?
My writing is very good. Interesting study. Good job. I just want to know, if you just push this. Given the cheetah and sambar density is so low, do you think that it can sustain such a high tiger leopard density? And is there a process of that prey is declining in carnivore?
So for Tadoba, we have seen that it has a sustainable prey density over the distributed over the study set.
It's very low, about if you put sulfur and cheetah together, about 5-6.
Yes, but when we compare it with the other study sites that we have seen that Tadoba is actually having the highest prey density among all the sites.
Thank you. Thank you. I will now invite Krishna.
Krishnan, your first question is from Ujjwal. I'll read it out to you. Okay, how did you establish paternity of cubs without genetic analysis, given you talk of faking paternity by females?
So the faking part came at the end of towards the end of the study. Before that, it was not present. So we can easily conclude that the dominant male present in that area was the father.
Next question is by Dr. He comments very interesting insights into the breeding biology of tigers. How would you explain very low cub survival of Maya female despite a strategy to mate with multiple males to create confusion about parentage? Is that a strategy he's asking?
Yeah. So again, strategy used by Maya came in the year 2023, and before that it was not utilized. So I don't think that was replicated later.
Okay, other questions are also related to paternity in absence of genetics. How we determine that? During tea time, people will, clarify that. I'll call on Anubhuti.
This is a question from Agyan. Interesting presentation. What was the prey status in the release site, release regions? As females need secured resources, low prey availability can escalate female versus female contest and influence dispersal. Please comment. And the second part is, have you considered analyzing the ectoderm data before and after dispersal?
So with regards to the first question, there is sufficient prey base within the release sites, and that is how the release sites were chosen to begin with in terms of where the release will take place. And the release sites for the two individuals are only 4 kilometers apart, so the local conditions for both the females were similar. So I don't believe that would be the case with this particular scenario. And as far as analyzing movement data for 21P and post dispersal is considered, we only got an opportunity to collar the individual at the time of the translocation. So we really don't have movement data prior to the translocation program itself. And that is also, this is very important because that would be one important strategy to consider for future translocations, because following individuals or looking at movement data of individuals before translocation could be an important indicator of how they may behave post translocation. So thank you for that.
Thank you, Anubhuti. Now, Ankit Thakur.
It's taking its time to focus. This is again a question by Kamar sir.
Given that approximate prey, cheetah size prey I'm talking about is about 3 per square kilometer in your area, what is the strategy for prey augmentation? That's the first one. And tiger carrying capacity seems to be overestimated.
you need to adjust for leopard and gold also. And the last one is that biomass-based equations are not appropriate because animals don't choose biomass; they choose animals. There's a lot of wastage also, so you need to account for that.
Do you have any comments with regard to Sir's comments or questions?
So yes, the first one is regarding Cheetal, right? Put together. Okay. So yes, I have a comment regarding Cheetal population. The population in the tiger reserve is restricted to certain pockets. It is clustered, and that too on the edges of the tiger reserve. And naturally, the population size is very low, and that's the reason I guess the number is on a negative end towards combining with sample as well. Second one is about the tiger carrying capacity. Yes, so for tiger carrying capacity, we basically presented all the two data sets that we had. So biomass predictive equation may be an overestimate, but since I was going through all the literatures that I've cited, I saw this predictive equation being used and being referred to multiple times. So I thought of coming up with both of the results, and maybe we can pick up the result of prey density based on the prey density.
One question only on this. You can pick any one, random choice.
Question is by Ayan. Which one do you want? First or second?
I'll make a choice first.
I was anyways prepared for that. So yes, it is something that we have actually discussed a lot about. But yes, point number one, the detection probability in Buxa Tiger Reserve due to the strong undergrowth is very low. Hence, we should be looking at the camera trap distance sampling as well. The only constraint is Buxa, the landscape of Buxa is not a uniform entity. So the northern part are very hilly regions, and although we are conducting a thorough systematic camera trapping exercise in that area as well, there is a limitation to the time factor that we can keep the camera traps. There are places where we need to
Ankit, so yes, now I hand over.
Okay. With respect to Buxa,
yes,
historically Buxa Tiger Reserve has always been a place where the apex predator has never been present. Are you working there with the assumption or with the thought that we will relocate tigers and make a tiger reserve where there were no tigers, we will bring tigers back? But historically, if you look from Buxa, it has never been a place for tigers. Tigers have been transient, coming and going. It will probably remain a habitat for other predators remaining there. So then, as a professional institution, you can say, well, the future for tiger is not good here. You may want to bring tigers, but the future of tiger is not here. Do you have that confidence to say so? Your work?
Yes. So about the historical context of tigers in Buxa, we have gone through certain literatures that have said, and coming on to one of the very recent findings and a study of Sarkar et al. In 2014 that came up with the DNA sampling based on the stat analysis, suggesting the tiger numbers to be around 4 to 15 individuals in 2014. So apart from our study, this has been the literature that we have reviewed, and I guess 14 to 15 tigers, we can't really say they being a transient one, even if you consider half of the probability of them being. So
Thank you. I invite Jaideep.
Jaideep, you have a question again from Ayan. refuses to focus on Iron's questions. So now, yes. So there is, Iron says there is a huge spatial shift in tiger distribution between 2020 and subsequent years. What are your comments on that?
Yes. So in 2020, due to the pandemic, we were able to cover 2/3 of the tiger reserve. So the area that is on the western edge of Nilgiri was not covered in that sampling period, which is why the temporal shift, the spatial shift shows that pattern.
Thank you, Jyoti. Now I'm going Baswaraj.
Swaraj, this question is by Dr. Samrat. Can you please elaborate your sampling strategy for the interview?
Dr. Samrat, there's a lot of space on this page. You know, next time write 3 columns.
I presume it was not random because your sample size is significant. I was wondering, have you selected these groups or individuals? Basically, he's asking you a question on your interview strategy and how did you conduct your sampling on that.
Thank you for the question, sir. Now we plug the data, we are treating it as a single landscape. We tried sampling all the sites. For example, for Koppal districts, we overlay 25 square kilometer grid and then we surveyed out of 292 total bits, we surveyed 228 grids. That was consuming lots of time. Over the period of time, we modified the sampling method in which we incorporated adaptive cluster sampling. So the initial selected grids were completely random. Once we visited those grids, if the respondent says that there is presence of animal, then we end up covering surrounding grids.
And the next question is by Dr. Bilal for presentation. Interesting work. Any specific reason for higher monsoon reporting of the species?
So this is the general pattern we have observed. It could be artifact of the data that we have collected, but since our detectors are humans, and humans are most of the times they are there in the field during monsoon, and yeah, that can be one of the plausible explanation for this.
Thank you, Pastor. I think he said that. Thank you. I now invite my co-chair, Dr. Salvador, for his comments.
Thank you, sir. Thank you, sir. It was a pleasure to listen to all the presenters, and I think we had 7 presentations, and a lot of them talked about interactions between carnivores, a lot of them talked about landscape planning, a lot of them talked about carrying capacity. So the overall impression of the presentations that I've got is that there's a gradual shift in the last decade or so, even more, especially when you look at Indian wildlife studies, especially if you look at studies in respect to carnivores, is that we moved from sort of behavioral observations to understanding patterns on a larger scale, understanding these patterns in terms of intricate behavior using modern technology. And I think that is the way things will progress in the field of, for example, ecology as well. We're also answering questions relating to carrying capacity on a larger conservation agenda. We want to really bring back species from their extinction and all of that. So these are the concepts that we are moving ahead, and it's great to see that a lot of the presenters have talked in that sense. Also in landscape planning and landscape heterogeneity, take those aspects into consideration. But all that is fine. What I would really want the presenters and their work to focus on is not to neglect the ecology of the species at the end of the day. All the models, all the technology can bring you that information. But at the end of the day, if you look closely at the ecology of the species, I think a lot of questions can be answered as to whether your model is saying something right or not. With that, I think it was a real pleasure to listen to all the speakers and for sticking to their time and answering the questions very well. Thank you, sir. Thank you.
I appreciate all the presenters that done an excellent job and reminded me when I was a researcher. The situation was different. We are using OHPs over a projector. And it will be a more or less a wrestling match between audience and the presenter. Actually, it is not something like that. I just want to tell one thing with my almost more than 30 years of experience, that statistics is the easy part. It doesn't take much. Natural history is very important, it's very hard. And what I'm observing over time, that we are slowly slipping into creating fancy models, which are statistically very interesting. They show out very beautiful maps. But in the end, what matters is good science. I'm not telling about this presentation only, presenters only, I'm in general telling that I think it's important for us to remain entrenched in basic ecology and natural history. It will go a long way. And that's my suggestion to my young colleagues, that make sure that whatever you do and present, it should be embedded in natural history of system and animals. And I end my talk by telling that beyond scientific insights and conservation strategy, I want to emphasize the broader significance of these deliberations. The philosophy that underscores our discussion is one of the reverence of our natural world. Our recognition that our well-being is massively linked to the system, the health of the system, and we should join hands to make sure that wildlife will survive. Unfortunately, our national parks are becoming glorified safaris, and we trust me, in a decade or so, we'll end up with just few national parks acting like zoos. We should make sure that wildlife should survive beyond protected areas. Once again, thank you, all of you, and I thank Dr. Shikha for assisting both of us, and to the audience. One more thing, that only few people are asking questions, and I want my young colleagues, feel free. I think the tradition of WII is not to judge researchers and students by asking questions, and I make sure, trust me, Director Sahib is also sitting there. If you ask difficult questions, you will not get reprimanded later on. So please ask questions. It is very crucial for development of science to ask critical questions in a nice way, in a very respectful way. Thank you.
Thank you, sir. We will now break for tea. Just an announcement for tea. The arrangements, due to limited space, have been made at two different places. At the auditorium, tea will be served for the guests, faculty, the presenters, and the project scientists and MSc students. And at the green lounge for the rest of the researchers and the staff. So I kindly invite you to break for tea, and we'll meet here back in 20 minutes. That's at 11:50. I request the next session's facilitator to kindly come and meet.
लाइव जाएगा लाइव नहीं मैं पहले का चार्ट तो इसी में देखा ना
बंद कर एक मिनट